Right, hi guys, welcome back. This is part 2 of our lecture about analysis of variance, our second lecture about analysis of variance, lecture 9 in the experimental design and analysis series of core skills. So before the break I told you about a non-parametric version of a one-way analysis of variance called Kruskal-Wallis. Now we're going to go back to thinking about the parametric analysis of variance, but we're going to look at more complex situations than we've done before, and specifically we're going to look at two-way analysis of variance, or two-factor analysis of variance. It gets called both of those things. And the objective here is really about understanding not just analysis of variance, but also a particular phenomenon that arises as soon as you've got more than one independent variable in an analysis, and that is the idea of a statistical interaction. So that's what we're going to focus on. So far with analysis of variance, and in fact all the other tests that we've looked at situations in which there's only one factor. So in the Darwin's-Finch's example from last week, I talked about a single factor that had 5 levels, and that was the species of finch in the study. And today, earlier on in the part 1 of this lecture, I talked about this wasp example where again we had 5 levels of one factor. The factor was the treatment applied to the males, and the 5 levels were various different opportunities or various different stimuli which might make the males want to copulate. So as I flagged, I think at the beginning of the last lecture, there are clearly lots of situations in which you want to do an experiment or a set of observations which is more complicated than this, where there is more than one factor in operation simultaneously. So the example I gave last week was this one where you might be looking at levels of pyrazine, which is this interesting molecule which is a defense chemical in ladybirds, and we might be interested in concentrations in 4 different life stages: eggs, larvae, pupae and adults, in 2 different species simultaneously. So we end up with 2 factors, life stage and species, being considered simultaneously in the same analysis. Now we can use analysis of variance for exactly this kind of problem, and it doesn't require us really to learn any new skills, any new concepts. It just exploits everything that we've already done. So we're going to look at that initially with an example from exercise physiology, and then it's such an important idea and it's quite difficult to get your head around the things that emerge from these more complicated experimental designs that we're going to look after the break at another example to try and really reinforce, in particular, this tricky concept of the statistical interaction. Okay, so one of the things that's kind of fashionable in the exercise world that you probably know about is this idea of high intensity interval training, where you can do short bursts, like really short bursts, 30 seconds of incredibly high intensity exercise, as high as you can go, and that this might be very beneficial physiologically, and there's some evidence that that's the case. So this is the example I'm going to talk about, comes from a recent paper looking at this so-called HIIT training, and whether it or other possible programs of physical training might be able to improve mitochondrial function in older people. So in older people, mitochondrial function is seen generally to decline, in the muscles in particular. And so here the study was looking at the mitochondria in the muscles and looking in particular at protein synthesis and looking to see if the age-related problems with protein synthesis might be reversible or fixable by giving elderly people particular forms of exercise training. So the experimental design was more complicated than the ones we've seen so far. There are 4 different exercise regimes. So we have sedentary people who are not being told to exercise at all, and then people are randomly assigned if they're not sedentary. If they're not randomly assigned to the sedentary group, they're randomly assigned to one of 3 different training regimes. There's the high intensity training, there's resistance training with weights, or there's a combination of different forms of training. I think a mixture of high intensity and resistance, but I can't be sure about that. But anyway, certainly some combined exercise regime. And the idea was to look at which, if any, of these regimes might improve mitochondrial function. But the key thing was we were interested in how older people in particular are affected by this, by exercise, and whether the general problems that we see with old people compared to young are rectified by the training. So it made sense to have both young and old people in the experiment, so we can get kind of baseline from what we'd expect in young people, and then we can compare it with what happens in the older people. So that was the design of the experiment. And if you think about it, this design gives rise really to 3 different null hypotheses about your response variable, which in this case was protein synthesis in the mitochondria. We've got young and old people in the trial, so one simple null hypothesis we could test is whether there's any overall difference in protein synthesis between old and young people. We've also got the 4 different exercise regimes or treatments, so we could look for differences among those. So we can test the null hypothesis that there is no overall difference between the exercise treatments. But then there's this additional hypothesis, which in this case is of interest to us, and that is that there'll be no difference in the effect of the exercise treatment on young and old people. In other words, what we mean is there might be some effect of the exercise, but is it different for young and old people? So for example, does high intensity exercise improve protein synthesis in old people more than it does in young people? That will be one example of that kind of question relating to this more complicated null hypothesis. And this third null hypothesis is the tricky one, and what we're describing here is the possibility that there might be a statistical interaction between the effect of exercise and the effect of age. So we're going to look at that as we go along. So here are some results, some data collected from these people, and we're looking at the synthesis rate of new protein per hour in the 4 different exercise regimes in young and old people. It's quite hard to see in a table what's going on, so let's bung it in a graph. This is actually the graph taken from the paper, and what we have here is a sort of slightly messy, complicated picture that's not immediately obvious what's going on. If you remember, we're trying to answer really 3 questions simultaneously relating to those 3 null hypotheses. Are young and old people different? So in order to figure that out, we need to compare the squares, the dark squares, with the open circles. So kind of ignoring this is the exercise regime down here. So you have to, in your mind's eye, try and ignore the exercise regime and just look at the dark squares versus the open circles. Is there evidence for higher protein synthesis in one age group or the other? It's not really very obvious. It looks like in the sedentary people, maybe the young people are doing better than the elderly people, but in at least some of the exercise regimes it's the other way around. Overall, it's a bit messy and hard to tell. What about the treatments? Are the treatments different? So here you have to try and ignore the difference between the young and the old and just ask, well, is there more protein synthesis in the sedentary people compared to, say, the high intensity or the other 2 exercise regimes? And again, it's not really very clear whether there is an effect or not. Obviously, we're going to need some stats to help us out here. What about that third question? Was the effect of the treatments different on young and old? So here we have to say, well, what's the effect on young people? Well, let's look at the open circles. So those, the filled squares. Filled squares are the younger people. The change in protein synthesis for them when you move from sedentary into high intensity exercise is very small, maybe a slight positive boost, but then it goes down a bit in the resistance training treatment, and it goes down a little bit in the combined treatment as well. So bit variable, not very clear for young people. What about old people? Well, the old people, they do seem to have done better moving from the sedentary condition into high intensity training, and possibly also when moving to resistance training, and possibly also when looking at combined training. So it looks like the impact of the training might be positive on the older people, whereas it wasn't so clear for the younger people. And that's the third question, really. We're trying to answer that third question. But all 3 questions, the answers are not completely clear-cut from the data, so we need to do some analysis to figure this out. So the approach here is going to be exactly the same as with one-way analysis of variance. The principle is we look at within-group variation and we compare it to among-group variation. In this case, we've got 8 groups in total. The within-group variation is going to be how much each individual person within a particular age group and a particular training regime varies, because not everybody's the same. Of course, there'll be random differences between individuals. And then the among-group variation is the stuff we're interested in. That's the stuff that is caused by maybe fundamental differences between young and old people, or effects of the training regime, or an interaction between the 2. So in order to sort of turn this into stats, we need to quantify that variation and put it into an analysis of variance table, an ANOVA table, like we did last week. I'm not going to look at any of the equations or anything, but it's the same principles that we used last week. So we want to fill up a table like this, but our situation is more complicated than last week. Last week we just had among subgroup variation and within subgroup variation. But this time life is more complicated. The variation among groups comes in three different flavours. We've got the variation which is going to be caused by differences in age, the variation caused by differences in treatment, and the variation caused by the interaction between those two things, the fact that the effect of the exercise treatment might be different on different age groups. So we're going to partition that among subgroup variation into three different sources of variation and then compare it with the remaining variation within the groups that's not related to the treatments or the age. So for all of these sources of variation, we can fill in this table. The computer will do it for you. What it's doing, remember, is measuring variation essentially in the form of sums of squares and then ultimately converting that into a statistic that we can evaluate with a p-value. So we've got essentially two forms of variation which map directly onto the factors that we're testing in our study, and then this additional form of variation which comes about because of the interaction between the two factors. And that's because it's a difficult thing to get your head around. I'm going to show you again another example after the break in the third part of the lecture. The final bit of variation in this table, originally I called it within subgroup. It's the within group variation, the random variation between individuals within a group. That, in these more complicated designs, and in fact in ANOVA in general, tends to get abbreviated to be the error term, the bit left over, or even the residual variation, because I've already taught you this word residual to mean the bit left over that's not explained by the things you're interested in. So not only is it called residual, it's also sometimes called error. And so this term on the bottom of an ANOVA table is typically called error. It's error because it's the bit that we're not interested in. It may actually be caused by measurement error. So the fact that when you're measuring metabolic rates or whatever it is, or protein synthesis, you're going to make mistakes, or you're going to have machines that don't quite give you a precise enough answer. But it can also, although it's called error, it can come about for all kinds of reasons, including interesting biological reasons, like maybe genetic differences among the people in your trials. The key thing is we don't know where it comes from, and for the purposes of this study, we're not really interested. We just want to make sure that we take it into account in the analysis. And the bottom line is, what we want to know is whether the differences caused by age or treatments or the interaction between them are greater than we'd expect by chance, given that every individual is different and therefore we have some noise, some error in the background. So stick it in a computer, the data in the computer, and you'll get an answer. You'll get a sum of squares for each of these things, reflecting the amount of variation, divided by the degrees of freedom gives you the mean square, and then crucially we can then calculate the test statistic F, which is the ratio of the mean squares for the thing you're interested in to the mean squares for the error. Why are we doing that? Well, basically what we're doing is we're asking how big is the variation among groups relative to the variation within groups, because if it's big, much bigger than we'd expect by chance, then that's going to suggest the null hypothesis is wrong. It's going to suggest that we're not just seeing the source of random variation between individuals here, we're seeing on top of that some real differences among age classes or among exercise treatments. How do we know exactly how likely these F values are to have occurred by chance? The answer, of course, is the p-value. That's the probability of seeing these differences between age classes, treatments, and the differences caused by the interaction under the null hypothesis. So here we have the results, and they can take some making sense of, so we're going to have a look at them in the context of the graph in a second, but it looks like we've got a non-significant effect of age, although it's quite close to being significant. We've got a non-significant effect of exercise treatment. At that point we might think, oh, we've found nothing interesting at all, but then we've got a really significant effect of the interaction, and that is going to need some unpicking. What does it mean? What have we actually found in this experiment? So let's just chuck away all the other stuff for the time being and focus on those p-values, which are telling us about how likely the data are under the null hypothesis. Should we reject those three null hypotheses that we started with? Let's look at them. The first one, So a non-significant effect of age, what does that mean? That means that if you look at this graph and ignore the exercise regime on the x-axis and just focus on the two different symbols, the cloud of black symbols, squares, is not significantly lower or higher than the cloud of open circles. So overall, we're not detecting a difference in protein synthesis between young and old people in this study. But that's not important, because remember, the reason we did the study was to find out whether the exercise made a difference to the potential disadvantage that elderly people suffer in terms of protein synthesis. Where is that disadvantage? Well, there's a hint of it here. Sedentary older people have lower protein synthesis than sedentary young people. And what we're interested in is whether exercise regime can change that. So far we don't know that. We just know that in this experiment overall there's no main difference between old and young people. The effect of treatment is also not significant. So what that's telling us is that if you ignore the different symbols now and just focus on the data for sedentary people versus the data on the high intensity treatment and the resistance training and the combined, there is no overall difference between these four different groups of data. So it's not like the protein synthesis rates are higher overall for the combined training or the sedentary or whatever. It looks like there are some differences, but they're not significant. But the thing we're really interested in is the interaction. And that's telling us that the effect of the exercise treatment is different for different ages, significantly different. Or putting it another way, the effect of age is different depending on which exercise regime the people were experiencing. Those two things mean the same thing. So let's kind of try and visualize that. If you focus, I've tried to add some arrows, I'm not sure if this really helps here, but if you look at the blue arrows show what happens when you move from sedentary in young people to resistance training or combined training. The answer is protein synthesis drops a bit. Contrast that with what happens when you go into those training regimes as an older person. If anything, your protein synthesis goes up quite a bit. So older people, when they're put on the resistance and the combined training, are getting better protein synthesis, and maybe also a bit with the high intensity. Whereas younger people, you're not seeing much change. The high intensity is actually the least clear-cut result, really. So it's easier to focus on the resistance and the combined training. For those two training regimes, it looks like younger people get lower protein synthesis, if anything, or maybe no change. Older people seem to improve. Putting that another way, if we look at the effect of age in each of the four different exercise regimes, it's actually a little bit easier to see this interaction. So what we're saying is, and this is exactly the same as what I showed you on the previous slide, but just put another way, that the difference between old and young people depends on the exercise regime. So in the sedentary condition, older people were a little bit lower than younger people, whereas in high intensity training it's about the same, and crucially in the resistance training and combined training there's quite a big increase in protein production for old people versus young people. So there we go, we have quite a complex conclusion from this paper that the exercise regime matters, but it matters in different ways for old and young people, and crucially it does seem to help old people, at least in terms of this one measure protein synthesis in the muscle. Whether that then gives rise to better muscle tone, better fitness and health in these people obviously is the subject of a further study, I'm sure. Okay, so that's an example of two-way ANOVA, two-factor ANOVA. Quite complex and confusing potentially, so we're going to look at another example after the break, and then we're going to finish off just by having a look at those nasty old assumptions that we have to worry about, and in particular the assumption of independence and what to do when it's violated.